Welcome friends. Today our guest speaker is Dr. Kritika Raj. She is a PhD in neuroscience from the Department of Genetics, University of Delhi, currently pursuing postdoc as an NPDF fellow grant from DST SERB from the Faculty of Life Sciences and Biotechnology, South Asian University. Dr. Kritika has long proven track record of working on neurodegenerative disorders in Drosophila models. Today, her topic of presentation is tissue-specific upregulation of Drosophila insulin receptor mitigates Poly Q-mediated neurotoxicity by restoration of cellular transcription machinery.
So thank you so much, Dr. Somnath, for inviting me to present my work over here, which has become essentially a platform for the most important platform, I would say, for communication and learning these days because of the pandemic situation, and we all are just sitting at home doing nothing. And you can connect to not just the scientific world, but also to fresh talent in terms of younger students who you can help learn through presentations like these, and you never know, they can get creative and do priceless things in science.
Right.
So, yeah.
Before we start, I have one quick question for you. What keeps you going in the field of research work?
I don't know. I always had this knack of doing something in science since my childhood. And, you know, I've always dreamt of being like having a professor job in one of the universities. So I think with that aim in my mind, I just keep going, and I just love going to the laboratory. You know, the research work that I do, it is a thing that I enjoy the most. So going to the lab, the smell of the chemicals, everything is so enticing, and I just enjoy, and that is what I think keeps me going all the time.
Right, I think it serves oxygen for you.
Yeah, you can say that.
Okay, so we can start with the presentation.
Okay, I will just start sharing the screen.
Yeah.
So what I'll be talking today about is majorly the work that I've carried out during my PhD tenure that entails the studies on the role of insulin signaling and its association with MYC in alleviating the human Poly Q-mediated neurodegeneration in Drosophila. I carried out this work, as I said, during my doctoral stint at the Department of Genetics, University of Delhi, which I completed in the year 2018, and currently, as Dr. Somnath introduced, I'm carrying out my postdoctoral fellowship at the Faculty of Life Sciences and Biotechnology at South Asian University. So, getting straight to the topic, as the topic suggests, this presentation is regarding polyglutamine disorders. So these are essentially a group of inherited neurodegenerative disorders that are caused by the expansion of the CAG trinucleotide in the coding region of the affected gene. Now, since the CAG trinucleotide encodes for the amino acid glutamine, therefore the protein that is made has a expanded Q stretch in it, and therefore the name Poly Q or polyglutamine disorders. Different types of polyglutamine disorders are various kinds of spinocerebellar ataxias, Huntington's disorders, DRPLA, etc. And the clinical features and symptoms that are commonly associated with these kinds of disorders are uncoordinated movements of hands and eyes, difficulty in maintaining gait and speech, cognitive thinking impairments, as well as memory loss. And in order to diagnose such disorders, the tool that is used worldwide is magnetic resonance imaging, or MRI. Some of the characteristic features of Poly Q disorders is their autosomal dominant pattern of inheritance, their late age onset and progressive course of development. And very interestingly, the severity of the disease directly correlates with the number of CAG repeats in the target gene. That is, more the number of CAG repeats, more severely will the disease manifest in the patient. So why do we study, why is there a need to study such polyglutamine disorders, and, you know, a need to combat such polyglutamine disorders? That is because they have significantly high incidences worldwide with devastating phenotypic manifestations. Until date, there is lack of mechanism-based strategies that are available in the market for treating such disorders. So there is an urgent need to find a cure. So this table summarizes all the genes, the wild type Q length, the pathogenic Q length, and the parts of the CNS that are affected in these different types of polyglutamine disorders. So the important thing to keep in mind over here is that all of these genes, irrespective of the polyglutamine disorder that they cause, have an intrinsic Q length in them, which is the wild type poly Q length, which when it gets expanded beyond a threshold level, then it causes pathogenicity. How? Let's see. So if we look at this particular schematic, if we take this to be a poly Q protein with the green box showing the poly Q stretch, it gets misfolded into a monomer that is rich in beta sheets. Now what happens is this misfolding renders this particular protein to become a substrate for active caspases and proteases in the cell, which leads to the production of misfolded fragments. These fragments associate with each other to form oligomers, which can further associate with each other to form inclusions, or what we generally call as inclusion bodies in the cell. All these different types of species that appear in the cell can lead to various kinds of toxic effects that can be transcriptional alterations, metabolic dysfunction, and proteasomal and stress response impairment. Now, these inclusion bodies can be of two types, either cytoplasmic in nature or nuclear in nature. However, it is the nuclear inclusions that are more toxic in the cell and that are more commonly found in most of the polyglutamine disorders that are known. For our studies, since the Huntington disorder and the spinocerebellar ataxia type 3 or SCA3 are the most widely occurring two types of polyglutamine disorders worldwide. So the studies were carried out on these two disorders using the Drosophila model system, which is a very well established model system for studying neurodegenerative disorders. So if we look at the life cycle of a Drosophila melanogaster in short, we can say that it has four major stages. One, the embryonic stage. Second is the larval stage, which can be divided into three stages: the first instar, second instar, and third instar stage, the pupal stage, and finally the adult stage. The entire life cycle takes about 12 days to complete at 25 degrees Celsius. And the model organ in the Drosophila that we chose for our studies was the Drosophila eye, because it is dispensable organ for the viability of the fly. Not indispensable, but dispensable. What do we mean by that? We mean that if anything happens to the eye of the fly, it does not affect the viability of the fly. And therefore, that is the beauty of using the eye as a model system. Plus, it is very easy to score phenotypes using the Drosophila eye, and it has both neuronal and non-neuronal tissues. So this is a virtual timeline of the Drosophila eye development. If we look over here, the third instar larva has a pair of these structures, which are known as the eye antennal disc, wherein this anterior portion forms the antenna, and the posterior, this particular portion, actually forms the eye of the adult eye of Drosophila, which looks like this. It is a compound eye made up of 700 individual units arranged in an array like this. And this individual unit is known as a ommatidium. Each ommatidium over here consists of 8 photoreceptor cells, or what we call as the rhabdomere cells, R1 to R8, and they're arranged in a flower-like arrangement like this. Now, in order to achieve targeted gene expression in the Drosophila system, we use a technique known as the US Galpo technique, wherein we make use of two independent transgenic lines. First is the enhancer trap Gal4 line, where the yeast transcription factor Gal4 is cloned downstream of a tissue-specific promoter. So here, since we are targeting the disease in the Drosophila eye, we will use a promoter that is specific to the eye of the Drosophila, which is the GMR promoter. And the other one is a US line, where we clone the transgene of interest downstream of the GAL4 binding US element. So essentially, these two lines independently do not express anything and are as good as wild type. But when these two flies are crossed together in the F1, it contains all the elements of this fly as well as this fly. So we achieve a tissue-specific activation of the transgene of interest based on the promoter-driven GAL4 expression, which then binds to the US element and therefore promotes the expression of the transgene. Now, in order to screen for modifiers against these polyglutamine disorders, this was the modifier screening strategy that was undertaken. So we took a US line, which has the gene of interest and has a normal looking eye like this. And we took a Poly Q expressing line, which shows a degenerated eye like this. Now, when these two kinds of flies are crossed together, three kinds of scenarios can appear. The first one is where the gene of interest acts like a positive modifier and rescues the eye phenotype, and therefore is called a rescue line. The second one, where the gene of interest acts like a negative modifier and exaggerates the disease phenotype beyond that of the disease. And the third one is the no effect. So we take ahead these kinds of lines and we discard these lines. Now, various players of the Drosophila insulin signaling pathway were analyzed for their effect as modifiers against polyglutamine disorders based on the hypothesis that the insulin signaling pathway is a major promoter of growth and metabolism in the cell, and therefore it can provide growth promoting signals to the dying polyglutamine or disease cells. Also, the insulin signaling pathway has been shown to be downregulated in most of these Poly Q conditions, and therefore we thought it would be a perfect fit to analyze pairs of these pathways for their modifier effect. So this is a simple schematic of the Drosophila insulin signaling pathway, where we represent the insulin receptor as INR. This binds to the Drosophila insulin-like peptides, gets autophosphorylated, and commences the downstream pathway that leads to modification of metabolism, growth, and proliferation. Some of the offshoots of this insulin signaling pathway can also be autophagy, translation, and endocytosis via S6 kinase. After extensive screening of players of the insulin signaling pathway for their effect against polyglutamine-induced neurotoxicity, it was found that the insulin receptor is capable of suppressing Poly Q-induced morphological deficits. And to start with, let me show you a schematic that gives you an idea about the percentage of identity between the wild-type Drosophila insulin receptor that is depicted by INR XL over here and the human insulin receptor. So as we can see here, there is 65% identity at the kinase domain, which is the catalytic domain of the present in the beta subunit of the receptor, and around 45 to 50% of identity in the A and B subunits, which are actually the ligand binding domain. So going on to the data, this is how a normal Drosophila eye looks like in wild-type condition. It is beautifully red in color and it shows a regular arrangement of ommatidia. However, when the Poly Q disease condition is targeted in the Drosophila eye, we see that the eye is completely degenerated. It is small in size. It is completely depigmented. The ommatidial lattice is disorganized, and we see what are these black small spots on the surface of the eye, which are indicative of necrotic cell death. However, when INR was co-expressed in this disease background, the disease condition was significantly restored. The eye size was reinstated, as were the pigments and the ommatidial lattice. Moreover, there was no appearance of any necrotic patch. On the other hand, when INR was downregulated using an INR RNAi approach, an equal and opposite effect was observed, validating the previous observation. Now, in order, these were light microscopy images. Now, in order to analyze the external surface of the eye in further detail, what we did was electron microscopy of the adult eye. So these are the electron microscopy pictures of the Drosophila adult eye. This is the wild type, which shows a beautiful array of ommatidia, and the bristle lattice also can be seen very well arranged. These are the high magnification images of the same. However, when the disease condition is induced in the eye, there is complete degeneration of the eye, and in fact, it collapses upon the critical drying step of electron microscopy, because there is lack of any internal tissue mass to support the shape of the eye. But when the INR is co-expressed in disease background, we see that the eye structure is reinstated with complete reformation of the ommatidial lattice as well as the bristle lattice. Now, after analyzing the external structure, we wanted to analyze the internal morphology of the Drosophila eye. So for that, we prepared horizontal sections of the Drosophila eye and brain, and subjected them to toluidine blue staining and thereafter light microscopy. So this is a schematic to give you an idea of how a horizontal section of the Drosophila eye and head looks like. So below the ommatidial facet lies an array of 3 ganglionic layers: the retina, the lamina, and the medulla, which is followed by the lobular portions, and then finally the brain. So this is depicted very nicely when a wild type section, when a section of the wild type fly is taken. As we can see here, below the ommatidial lattice, the retina is very beautifully placed, followed by the lamina. However, in case of disease flies, this eye is completely degenerated with absolute loss of the internal retinal structures. However, when INR is co-expressed in disease condition, we see that the morphological deficits are significantly improved, and the internal retinal mass is also restored. Now, since the polyglutamine disorders, as I explained earlier, these are age-related disorders, and the severity of the disease tends to worsen with the increased age of the person. So therefore, it was important to analyze the modifier effect of INR with growing age of the flies. So as we can see here, when Poly Q disease condition is induced, the disease condition gradually worsens with age, and at 20 days post-eclosion, that is when the fly is 20 days old, we see a completely degenerated eye with large necrotic patches that can be observed on the surface of the eye. However, when INR is overexpressed, it is able to rescue the eye phenotype even 20 days post-eclosion, and the necrotic patches are not there, that is there, but even the pigments and everything is completely is there to a significant level even 20 days post-eclosion. Now, the rescue potential for INR was also analyzed using other models of polyglutamine disorders. The first one was by utilizing a moderate form of spinocerebellar ataxia type 3. This transgenic line does not produce any visible morphological deficits when we look at the eye of the fly per se, but upon subjecting this eye to deep pseudopupil analysis, which gives a readout of these photoreceptor cells inside each ommatidium, we can see a difference. How? So let me just explain to you what a deep pseudopupil analysis tells us. So as I explained earlier, the Drosophila adult eye is made up of 700 ommatidia, and each ommatidia has this arrangement of rhabdomere cells inside them. Now, since the rhabdomere 7 and 8 are overlapping in nature, only one of these cells can be observed at one plane when we do the microscopy. So that is what is shown over here in wild type cells. In wild type flies, upon a deep pseudopupil analysis, we can see 1, 2, 3, 4, 5, 6, 7. 7 rhabdomere cells beautifully arranged in this flower-like arrangement inside each ommatidia. This condition is completely degenerated in a disease-expressing fly, although it does not show any morphological deficits over here. But the photoreceptor cells are completely degenerated with an average of only 1 or 2 photoreceptor cells that is visible in each ommatidium. However, when INR is overexpressed in such a background, we see a significant improvement in the number of photoreceptor cells, and there is an average of 5 to 6 photoreceptor cells that is visible in each ommatidia. And similar results were also obtained when toluidine blue staining was carried out in horizontal sections of the adult eye and brain. And the second line that was used to study the effect of INR was a Huntington line, where there are 138 Q repeats in the Huntington gene, and similar kind of rescue results were obtained. And so was that when the studies was carried out with another Huntington line, which contains 93 Q repeats. So now, the results rescue was obtained, everything is going fine. But how do we analyze that indeed the rescue effect that is being achieved is indeed because of the overexpression of INR? So for that, we carried out immunostaining against the insulin receptor, and these are all confocal microscopy images after subjecting the third instar larval eye disc to immunostaining against INR. So this is the wild type. This is in wild type condition. We can see an expression of the insulin receptor in the area posterior to the morphogenetic furrow in this posterior region, which will make the eye, as I explained earlier. And this expression was particularly restricted to the boundaries of the cell. That is very typical of a receptor protein. Now, in case of disease condition, this expression was slightly reduced. And however, when INR was overexpressed, we see a beautiful overexpression of INR in the eye region, which is more clearly observable here, which clearly indicates that yes, INR is actually being overexpressed in these cells, and therefore the rescue effect is indeed because of the overexpression of INR. And this is the quantitative analysis of the same. Then, in order to validate the rescue potency of INR, an independent another allele of INR was used, which has a cyan fluorescent protein tag downstream of it. So when this kind of transgenic line was used, although wild type and disease condition discs do not show any kind of cyan fluorescence in the eye region, so these are all fluorescent images, fluorescent microscopy images. But when the third instar larval eye disc, which contain the INR CFP in Poly Q background, was subjected to fluorescent analysis, we see a beautiful expression of the cyan fluorescent protein in the region, in the eye specific region, which shows that yes, INR CFP is being expressed. Now, when these flies were analyzed as adults, we could also see a rescue of the disease condition in these flies, thereby indicating that yes, this particular allele can also bring about rescue of the disease phenotype. Now, in order to validate that yes, the INR expression is being achieved in these adult eyes, and the rescue event is indeed because of the expression of this INR CFP, the adult heads were subjected to fluorescent microscopy, and we could see a beautiful expression of a beautiful CFP expression in the eye region of the adult Drosophila head. Now, moving on to the architectural deficits and the functional deficits that Poly Q caused, it was imperative to study the potency of INR in rescuing such deficits. For that, first of all, phalloidin tritri staining was carried out, which stains the F-actin and therefore stains the boundaries of photoreceptor cells and the photoreceptor pigments present at the center of these cells. So here in wild type condition, we can see a very beautifully regularly arranged array of photoreceptor cells and photoreceptor pigments at the center of the cells. However, when upon staining in disease conditions, we see a very dysregulated and disoriented lattice with gaps somewhere between the ommatidia and somewhere the lack of photoreceptor pigments at the center of the ommatidia. When INR is overexpressed in such a background, it leads to reinstation of the disease phenotypes and therefore rescues the architectural deficit. Now, in order to study the functional rescue, we do what is known as a Y-maze assay. Here we take a test tube-like apparatus, which is shaped in the form of a Y letter, and the two arms of the Y are treated differently. One is illuminated and the other one is the dark arm. So the rationale of this assay is that flies with normal vision will always walk towards the illuminated arm of the Y-maze, whereas the flies with compromised vision will walk 50-50 towards the dark arm and the light arm. So when these three kinds of flies were subjected to Y-maze analysis, we saw that the wild type flies moved towards the light arm, but the disease flies, which moved 50-50% in the light and the dark arm, which suggests that there was a functional deficit in these flies. Half of the flies were blind, they could not see, and therefore half of them moved towards the dark arm. The third one, this one shows when INR was overexpressed in the Poly Q background. And here we see an increase in the number of flies that are moving towards the light arm, which suggests a functional rescue facilitated by INR. Now, since Poly Q disorders are brain-related disorders, it was very essential to study the suppressor effect of INR against Poly Q-induced neurodegeneration in the central nervous system. For this, we used two different drivers. One is the ELAV-GAL4 driver, which drives the Poly Q condition pan-neuronally in the central nervous system of the fly. And the other one was the 201Y-GAL4 driver, which drives the disease condition in the mushroom body of the Drosophila head, of the Drosophila brain. So the mushroom body is actually the Drosophila counterpart of the human hippocampus and is responsible for learning and memory, which are two of the most important traits that are affected in these types of disorders. So the mushroom body is essentially an L-shaped structure, as we can see over here, with prominent and distinct alpha, beta, and gamma lobes. And this kind of a structure can be easily stained with Fas2 staining that stains the axons of the mushroom body neurons. So when Fas2 staining was carried out in ELAV-driven and 201Y-GAL4-driven flies, we saw very beautifully stained mushroom bodies with distinct alpha, beta, and gamma lobes. However, in case of disease condition with both the drivers, not only was the staining intensity reduced, but there were various abnormalities of the mushroom bodies that were identified, namely the reduced alpha arms, the distorted beta arms, and a complete absence of gamma arms in some of the cases, which indicates towards their complete degeneration. However, when INR was overexpressed in such backgrounds, we saw significant improvement in mushroom body morphology as well as the staining intensity. And this is a graph that shows the length of the mushroom bodies and in these different kinds of phenotypes. Now, also, since polyglutamine disorders manifest due to extensive cell death and tissue loss, it was important to study the suppressor effect of Poly Q against such apoptotic and cellular stress events. So for that, first of all, the tunnel assay was carried out in third instar larval and adult head sections. In wild type conditions, we see a very minimal apoptotic cell death, which is also the basal level that is expected in any kind of tissue. However, in the disease condition, we observed apoptotic cell indicative tunnel puncta very profusely distributed in the eye tissue. However, when INR was overexpressed, this condition was significantly improved. Apoptosis was inhibited, and there were only very minimal uh, um, uh, apoptotic puncta observed again. Now, cellular stress and apoptosis was also measured using staining against other markers for these conditions, such as activity neuron staining, cleaved caspase staining, and HSP70 staining, and similar results were obtained both qualitatively as well as quantitatively. Now, coming to the most important part, as we had talked about that the formation of protein aggregates is a major hallmark of these kinds of disorders. Therefore, it was important to check what is the effect of INR that it has upon the formation of these kinds of protein aggregates. For this, we used two kinds of lines. First one was the SCA3 severe line, which has a Q78 stretch and has an HA tag at the N terminus of the pathogenic ataxin 3 gene. The other one was the Huntington line, which has a 138Q repeat and has an MRFP tag at the C terminus of the pathogenic Huntington gene. So in the first case, where there's an HA tag upstream of the ataxin gene, the tissues were subjected to staining against the HA tag. In case of the disease flies, we see abundant presence of protein aggregates in the eye region of the larval iris. And these aggregates, protein aggregates, were majorly nuclear in nature, as we can see over here by the yellow signals which occurs by the overlap of the red and green signals, and these were also fairly big in size. However, when INR was overexpressed in these kind of flies, the abundance of protein aggregates was significantly reduced, as is visible over here in the eye region. The aggregates were significantly reduced, and whatever were there, the aggregates, whatever less were there, were also cytoplasmic in nature and very small in size. This is the quantitative analysis of the same. And when a Western blot was run for these conditions in the disease fly, we see that the protein aggregates were SDS insoluble and remained trapped in the stacking gel. However, in case of INR overexpression in disease background, the intensity of the band of the stacking gel was significantly reduced, and there was an equal and opposite intensification of the band over here in the resolving gel, which points towards the fact that maybe INR is working by breaking down these SDS insoluble aggregates into smaller species, which can be oligomers, and thereby bringing about the rescue event. Now, with the second line, which was the Huntington line, having an RFP tag downstream of the Huntington gene, because of this RFP tag, it was easy to do in vivo analysis in these flies, and we directly did fluorescent microscopy by isolating the third instar larval iris from the disease and the INR overexpressed flies, and we subjected them to RFP analysis. And here we see that there is profuse abundance of protein aggregates in the eye region that can be seen in the disease flies, which is significantly reduced in case of INR overexpression. And when similar analysis was done using the adult heads, a similar kind of result was obtained with profuse abundance of aggregates in the eye region of the adult heads in disease flies, and the restoration or inhibition of these kind of aggregates when INR was overexpressed. Now, we saw all this data. We know that the INR is overexpression of INR is inhibiting disease phenotype morphologically and functionally. Now we wanted to assess the mechanism by which this event is happening. And since we last talked about the rescue or the inhibition of aggregate formation by INR overexpression, the first mechanism that was assessed was that of agriphagy, which is a specialized autophagy mechanism that targets the protein aggregates in the cell. So also, because autophagy is one of the most important pathways that is downregulated in case of most of the Poly Q disorders, and it is also one of the pathways, one of the well-known downstream pathways of the insulin signaling pathway. So it was important to check the effect of autophagy in the cell. So for that, we used a lysotracker red dye and we did fluorescent microscopy. What this lysotracker red dye does is that it stains active lysosomes in the cell, and therefore it gives a readout of the active autophagy in the cell. So this is the condition in wild type case where there's a basal level of autophagy being carried out in the cell, which is downregulated in case of disease condition, which very well corroborated with the previous literature. And then when INR was overexpressed in disease background, we see significant induction of the agriphagy or autophagy processes in the eye region of the larval iris. Similarly, we also used LC3 staining, which is a staining for an LC3 molecule that is an important molecule present on the membrane of the autophagosomes, and it also gives a readout of the autophagic process in the cell, and a similar kind of result was obtained both quantitatively and also by Western blotting. Now, the second mechanism that was analyzed was that of cellular growth versus cellular proliferation. This was because the insulin signaling pathway is a potent inducer of both cellular growth and proliferation in the cell. So it was important to segregate which of the two mechanisms is being followed in order to achieve the rescue event. Is it the replenishment of the dying neuronal cells by their re-entry into the cell cycle and leading to proliferation, or it is just by cellular growth? So for that, we carried out anti-BRD staining, which stains the actively dividing cells in the tissue. So as we can see here in the wild type condition, there is a very beautiful BRD staining observed at the morphogenetic furrow of the iris, which contains all the actively dividing cells. However, in case of disease condition, we saw discrepancies in the BRDU incorporation in the morphogenetic furrow that points towards the degeneration of cells and loss of proliferation of cells. In case of INR overexpression, we see that the status of proliferation of cells was restored, but it was not more than that observed in the wild type, which suggests that INR is able to rescue the dying cells, but it is not because of overproliferation of the cells, because it is not exceeding the wild type levels, and therefore it is only because of the growth promoting properties of INR. In order to further elaborate upon this aspect of rescue mechanism, we used 3 different transgenic lines of insulin receptor in addition to the wild type line that we were using, which is this INR XL transgenic line, which expresses the wild type insulin receptor. The first transgenic line that was used was the INR A1325B line, which has an A to D mutation at position number 1325 in the transmembrane region of the beta subunit. This particular transgenic line was capable of inducing both signaling and growth of the insulin mediated by the insulin receptor, irrespective of this particular mutation. The second line was an INR R418P line, which carried a point mutation, R to P point mutation at position number 418 in the alpha subunit. This particular mutant line, although it was capable of constitutive signaling by the insulin receptor, it inhibited growth properties of the cell. And the fourth line was the INR del, in which most of the N-terminal region of the insulin receptor was deleted. And this line was also capable of constitutive signaling, but inhibited growth. So now, in comparison to the INR cell, so here it is the wild type INR that is overexpressed in disease background and which shows this kind of rescue. It was only the INR A1325D mutant that was able to bring about rescue of the eye phenotype, and neither was R418P able to do that, nor did INR del bring about such rescue, which suggests that the growth promoting properties which are lacking in these two kinds of mutants is important in INR. And this is a schematic that shows the similar kind of effect even with growing age of the fly. Now, the third and the most important mechanism that was looked into was that of the transcriptional dysregulation. Another aspect of polyglutamine disorders is transcriptional dysregulation, which happens because of sequestration of major transcription factors in the polyglutamine containing protein aggregates. And CBP is also one of the transcription factors. As we all know, it is one of the most important transcription factors in the cell, and it is also one of the transcription factors that is sequestered in the protein aggregates by way of homotypic glutamine interactions that happen by this polyglutamine stretch that is intrinsically there in the CBP protein. Now, since CBP is also a very well-known downstream effector of the insulin signaling pathway, we sought to determine the effect of CBP modulations in the rescue mechanism. So when CBP alone is overexpressed in disease background, there is no significant rescue that is observed. But when CBP is expressed in addition to INR in the disease background, we see an additional rescue, an additive rescue over here with completely normal looking eye that can be observed, and this eye was actually solid, which points towards the presence of even internal structures that have been replaced in such eyes, in such flies, sorry. And when CBP alone or CBP in conjunction was with INR was expressed in the eyes without any disease condition, we see an increased eye size in both these conditions. Sorry, when CBP overexpressed and INR and CBP both, with even ommatidial fusions seen on the surface of the eyes, which is because of the overexpression of two most important and most strong growth promoting factors that are being expressed over here. Both CBP and INR, as we know, are strong growth promoting factors, because of which it causes the eye to have an increased size and also leads to ommatidial fusion. Now, following this, genetic interaction studies were carried out using 3 different lines of CBP, transgenic lines. The first one was the CBP FLAD, which had a dominant negative mutation in this histone acetyltransferase domain. As we all know, CBP is a very potent histone acetyltransferase in the cell. So the FLAD had a dominant negative mutation. The CBP delta BHQ was deleted for the histone acetyltransferase domain, and the last one was the CBP RNAi. So when these three kinds of transgenics were expressed in the Drosophila eye, they themselves caused degenerated eye phenotypes because there was expression of a mutant CBP, a mutant transcription factor. When these kinds of mutants were expressed in addition to the disease condition, it led to an eye phenotype that was worse than that of the disease condition. It led to the exaggeration of the disease phenotype because there were two additive factors working over here: the disease condition plus a mutant transcription factor. However, when INR was overexpressed in these backgrounds, which had a mutant CBP and Poly Q, when INR was overexpressed in such conditions, some rescue was observed in this kind of fly which had the CBP FLAD mutant, and no rescue was observed in the other two transgenic lines, which suggests that CBP works downstream of INR in order to bring about the rescue event. Because here, when the function of CBP is abrogated, this is completely deleted of the acetyltransferase domain, and this is an RNAi mutant. They do not bring about any rescue, because even if INR is working upstream, if the downstream effector is absent, no rescue event is observed. However, in this one, some rescue is observed because this is just a dominant negative mutant, which means some portion of CBP is still working in the cell. Now, in order to validate that, yes, CBP does work downstream of the INR to suppress Poly Q-induced toxicity, we again carried out immunostaining against CBP and carried out confocal microscopy. And we could see that in wild type conditions, this is the basal level of CBP expression in the cell, the transcription, which suggests the transcriptional state. And in disease condition, it is completely downregulated, which points towards the transcriptional dysregulation. And when INR is overexpressed in the disease background, we see a very beautifully expressing CBP in the eye region, and therefore validating that CBP is working downstream of INR in order to bring about the rescue event. Now, since CBP is a histone acetyltransferase, it was important to check the status of the abundance of Poly Q-induced heterochromatin in the three models that we are using here. So for that, we checked the status of methylation and acetylation of lysine position 9 of histone H3, because this is one of the most commonly modified residue of the histone H3. So when we carried out immunostaining against the acetyl K9 of histone H3, this is the basal level acetylation status that was observed. However, in case of disease condition, there was almost no staining observed, which points towards the transcriptional dysregulation or the abundance of Poly Q-induced heterochromatin in these tissues. However, when INR was overexpressed in the disease background, we see that the transcriptional dysregulation is inhibited, and the acetylation is promoted, and thereby leading to the inhibition of heterochromatic conditions, which was Poly Q-induced. And this is the high magnification images of the same, the quantitative analysis of the same, and Western blot analysis. The same thing was also analyzed with the methylation status, and it gave an equal and opposite result, therefore validating the previous observations. So therefore, insulin signaling or INR was found to restore the histone acetylation levels and found to moderate the abundance of heterochromatin and transcriptional dysregulation, which was Poly Q-induced. Now, since the first step in insulin signaling is the phosphorylation of the insulin receptor, and since we are proposing that the rescue event is happening by way of the activation of insulin signaling, which leads to the activation of CBP downstream of it, and thereby bringing about the rescue event by transcriptional modifications, we sought to further analyze whether or not the signaling pathway is indeed getting activated. And the first step, as I said, is the phosphorylation of the receptor. We carried out immunostaining against the phosphorylated state of the insulin receptor. These are again confocal images of the third instar larval eye disc subjected to immunostaining against phospho-INR. And in wild type conditions, there is just a basal level of phospho-INR staining that is observed here, and which is slightly increased in case of the disease flies, which can be attributed towards basal stress response against these kind of disorders. And then when INR is overexpressed, we see profuse expression or profuse staining of the phospho state of the insulin receptor, pointing towards the presence of phosphorylated species of the insulin receptor in such conditions, thereby pointing towards the activated state of the insulin signaling pathway. Now, genetic interaction studies were carried out to further analyze this particular aspect of the signaling, and this was carried out by doing light microscopy for the eyes and for deep pseudopupil analysis for the same flies. This is the wild type, this is the disease condition, this is with INR overexpression. So this is in short the flow of the insulin signaling pathway. So this case, where shows the down regulation of the PI3 kinase, which is over here. So when PI3 kinase is down regulated, no rescue is seen because one of the players of the insulin signaling pathway is absent, is being taken away from the signaling. This condition where AKT is upregulated here, when this AKT player of the signaling is upregulated, again a rescue is seen. Then when SGG, which is the Drosophila homologue of GSK3 beta, when it is upregulated, there is no rescue that is observed and a degenerated eye is observed. Why? Because GSK3 beta is negatively regulated by the insulin signaling pathway. So when this is upregulated, no rescue is observed. When it is downregulated, as it is here, some rescue is observed. Now here, when INR is upregulated, but CBP is downregulated, the CBP, which is the downstream effector, no rescue is seen because the INR, although is giving the signal, but there is no downstream molecule to effector molecule to respond to that signal. So therefore, no effect. Again, when MYC is upregulated, a rescue event is seen, and when INR and MYC both are upregulated, an additive rescue is seen. Similar were the results that were obtained with deep pseudopupil analysis, and similar results were also observed upon genetic interaction studies with an HD disease model, that is a Huntington's disease model, thereby validating our results for the insulin signaling pathway analysis. So finally, the model that was proposed as a result of all of these studies was that insulin receptor works to restore or mitigate Poly Q-induced neurodegeneration by enhanced transcription in the cell, which can lead to either inhibition of protein aggregation or their their breakdown into simpler molecules or monomolecules. This enhanced transcription can also lead to or induce autophagy or specialized agriphagy in the cell. It can also lead to increased CBP protein in the cell, leading to transcriptional restoration, and it can also lead to inhibition of cellular stress and apoptosis. Now, the future prospect and the present work that I'm doing in my postdoctoral work right now is to validate these studies in cell culture systems and in systems in neuronal cells that are obtained from mouse embryonic tissues, because as long as we do not validate such studies in human cell lines, the data cannot be really validated. So I'm using different kinds of cell lines like HEK, Neuro2A, and trying to prepare stable cell lines where I can do transfections with the mutant form of ataxin-3 and the insulin receptor, and study the effects on cellular morphology and see whether the insulin receptor has a similar kind of rescue effect in cell culture systems also. And then finally, I will also be, I also plan to do similar experiments by isolating neuronal cells from my mouse embryonic tissues and carrying out the similar kind of experiments and validating my modifiers. The publications that I have brought out from this particular work is this, that was accepted in 2018 and was online in 2019. And I have also been lucky to be, for my work to be highlighted by some of the print media and online media, one of the leading newspapers also, like the Hindu, etc., and the online platforms. And this is the list of publications that I have. And finally, I would like to end with an acknowledgement slide. I would like to thank my PhD supervisor, Dr. Surajit Sarkar, and my present postdoc supervisor, Dr. Ravi Shankar Sundhi, for believing in me and supporting me with my work in every way possible. And I would also like to thank Department of Genetics, Delhi University, FLSB, South Asian University, all the funding agencies, and all the scientists in India and abroad who have helped me with Drosophila stocks to carry out my work and make me reach where I am today. Thank you.
Thank you, Dr. Kritika. It was a wonderful talk. I thoroughly enjoyed it, and I was so engrossed into those lovely images that I didn't feel like stopping in between.
I thought this work would be a perfect fit to be presented on a channel like this, because the aim of this channel is also, I think, is focuses on data acquisition and analysis of this sort, like various kinds of imaging techniques. And since this work involved extensive imaging techniques in terms of fluorescent microscopy, confocal microscopy, light microscopy, all kinds of microscopy, I thought it would be a perfect fit to be exhibited on a channel like this.
Very true, very true. I think I've never seen a much apt talk in my platform yet, which suits with so much microscopy images. But yeah. I have few questions. So I could see that the entire pathway of how you have started from an insulin receptor overexpressing it, and then going all the way down to like the CBP regulations, histone modifications. So it was really wonderful to see how they like the up and down regulations and how the rescue is being done. But my question is, like, see, if this is the entire pathway which you have studied or which you have followed, like how many sets of animal models which you have like initially designed for your plan of study, or with due course of time you thought of going ahead with the like for example C-AMP binding proteins. So you wanted to go ahead with that after certain movement in your work, or you had pre-planned it right at the beginning?
So to answer the first part of your question, actually, we procure these kinds of transgenic lines from the Bloomington Stock Center that is there in USA. So these stock centers have already prepared these kind of transgenic lines, and you can directly procure these kinds of lines from them. Some of these lines we have also produced in the lab, like for example, this particular line, which is a constitutively Poly Q expressing line. So this we have produced in the lab in-house by crossing them with balancer lines such as Curlio, so that it gives you also a marker to identify with the disease and rescue flies. So some of these lines are produced in lab by simple genetic crosses. And for the later part of the question, so I actually did not show that in my presentation. I skipped that part. This part was the genesis of this work was actually based on a previous study from the lab, which identified the Drosophila mic as a modifier of such polyglutamine disorders. And there also we had identified MIC to be rescuing the disease phenotype via the CBP modulation.
Thank you, Dr. Kritika. It was really a pleasure listening to your talk. And but I have some, like as I said, I have a lot of questions, but I will leave it to the viewers also. They can comment below and they can always get back with answers. My question is, you have used a lot of confocal microscope images, and they were really beautiful. I could see the ommatidia in them, and it could be like countable. That is the most important thing. So what systems you used for
so these
imaging?
are for confocal imaging. We have used the Leica SP5, I think 2, SP5 model number 2. It said. So that is the confocal microscope that we have used, and we have also done the offline editing and all on the Leica microscope that was there, that was available in the central instrumentation facility at the Department of Genetics.
Right, right. Okay, and while working with Drosophila, I know it is not as easy as you see in the pictures.
I think I now feel, as compared to cell culture systems, Drosophila is a much easier model system to handle.
So you have your hands set.
Systems are quite tricky.
Right, right. So I have like the images when you were showing me the ommatidia, I could see them count. I could count them actually, and so it is usually very difficult when you put the Drosophila head. Either you use the agar bed to mount the head in a proper orientation, and after that you do a lot of Z-stack so that you get all the planes, because it is not a flat eye.
Yeah,
so you used any special tricks to get those images?
No. So it is a very simple technique, but you just need to get your hands on it, because these deep pseudo people images were captured at high magnifications, and for that we just took the Drosophila head, we put a drop of oil on it, and we simply did light microscopy for the heads at high magnification 60x. These were 60x images. You could also use 100x depending on the objective of the experiment.
Right.
And these were done by reducing the aperture of the light that was falling on the head. So a very thin beam of light was made to fall on the head, and this was just so one eye of the head was focused at one point, and out of that also a very thin beam, the aperture was reduced to a minimum and then focused at one particular set of ommatidia at one time. So it is a little tricky, because in oil the head tends to move when you move the stage of the microscope, and it does not stay at one particular point for you to be able to capture the image. But it's just again a hands-on game. You have to just set your hands, and with the amount of oil that you use and how you do set the plane of the head. So it's nothing nothing fancy over there. You just put the head horizontally on the slide, put a drop of oil, and do light microscopy.
Okay, thank you. Because there are a lot of people who are working on a fly lab, and they usually face this problem. So I just wanted to have it from you, how best tricks or any tips for them.
I have actually, most of the people have after this presentation have asked me about this very technique only, and I tried to explain it here, but if in case somebody wants to further learn, they can contact me on the ID that I've given, and on any other Instagram or anywhere else that they can catch hold of me.
Sure, sure.
Or in the comment section.
Yeah, sure. I would request the viewers also, please, you should see this entire talk, and not only see, take a take-home message from here and imply in your lab work. If you have any questions, please feel free to comment below. Dr. Kritika is always there to help you out.
Happy to answer.
Yeah, happy to answer. So please, see this video, and then I'm looking forward to your comments and your practical problems which you have been facing. We would be more than happy to help you out in all possible ways. Thank you. With this, I conclude today's session. I would again thank Dr. Kritika for a wonderful talk. Thank you.
Thank you so much. Thank you. Thank you for having me.
Thank you.